I'd like to specifically say thank you very much to Daniel for organizing this and for also playing a pivotal role in bringing MEG to Sweden. As he mentioned, I'm going to be talking about NeuroSQUID, which is the new project that we have funded thanks to the Newton Alice Wallenberry Foundation. And I'll start by talking a little bit about why one might want to do MEG with high TC SQUIDs. Then I'll move on to what we have done previously with 1 and 2 channel MEG recordings with our SQUIDs. And then finally, I'll talk about what our future plans are and what we hope to do with this NeuroSQUID project. Of course, it's critical to point out, as has already been mentioned, this NeuroSQUID project is thanks in very large part to the Newton Alice Wallenberry Foundation. But we've also been receiving funding from Vetenskapsrådet, and this is a sort of spin-off from a previous FP7 project called MEG-MRI that was hosted by colleagues in Helsinki. So, to start with MEG and high TC SQUIDs, most of you probably already know a lot of this, but I understand that there may be some people that perhaps are more on the sensor side, so I'm going to be providing a very rough overview of what MEG is. So those of you that already know, you can probably just take a nap. But obviously, magnetoencephalography is a mouthful, so most of the time I'll be referring to it as MEG. It stands for magneto, so magnetic fields, encephalo being the brain, and graphy recording. And basically, this is what magnetoencephalography systems look like today. So you have a large dewar housing a set of sensors that are recording magnetic fields on a subject sitting in a chair or lying down. What is it used for? Well, as Daniel already very clearly pointed out, so there's lots of functional neuroimaging investigations that can be done with magnetoencephalography. The main benefits are the fact that it's studying electrical activity in the brain, which means it can have very high temporal resolution. I'll talk more about that in a moment. And it's a passive and safe functional neuroimaging method. But I think more generally speaking, it's important to put things into context in terms of why studying the brain is a critical thing for our future. And that's coming from the fact that if we look at the total cost of brain disorders, so this is anything clinical or psychiatric related, anytime something's sort of going wrong with the brain, there's an estimate in 2010 that roughly 800 billion euros of cost went to taking care of brain disorders of different kinds. And this is already 5 years ago. We know this cost is going to grow, perhaps most importantly because of the social and demographic changes happening. As the population continues to age, we know diseases of the brain are going to become increasingly costly for Europe as well as the rest of the world. So understanding the brain with functional neuroimaging techniques is one of the ways in which we can try to alleviate this cost in the future, this growing burden. So what is it that's special about MEG compared to other neuroimaging techniques? This is again, well, I should point out, this is a very biased comparison and it's quite hand-wavy, so physicists may not like this so much, but I think this is a good way of sort of presenting how one might compare the different types of techniques that people use today for comparing neuroimaging. So basically, you have a set of techniques that image chemical and metabolic neural activity. And these are fundamentally limited in temporal resolution to something on the order of 1 second. And that's not really because of the hardware. Many of these techniques, for example, FNIRS, you can sample at kilohertz frequencies if you want. But you don't gain any more information about the brain activity itself because of the fact that you're imaging chemical and metabolic activity that's being modified inside the brain basically by blood flow and other slow dynamic processes that occur at time scales on the order of 1 second. So even if you could, for example, build an fMRI system that could sample down into the millisecond scale, you wouldn't be gaining more information about the functional activity in the brain because fMRI is looking at blood oxygenation. Yeah? So the other types look at electrical neuroactivity. So you have EEG and MEG. EEG is looking at electric fields. You basically just put electrodes around the head and measure. You can have what's called HD EEG, where you place many more electrodes than the standard, let's say. And with those techniques, you get a fairly large range and spatial resolution, as well as a somewhat limited temporal resolution as well. So this crossover, let's say, is quite vague, but it's essentially limited and lower, or not as good, for example, as the state-of-the-art in MEG. So this is one of the things that's really powerful about MEG. It has extremely high temporal resolution. It's imaging changes in brain function that are visible on the order of milliseconds. But what is it that we're actually looking at when we study electrical activity in the brain? Well, to get a measurable field outside of the surface of the head, so this is a passive and non-invasive technique, you need lots of, so something on the order of thousands of neurons that are stimulated semi-synchronously. And you need those neurons to be a special type called pyramidal cells, and their dendrites have to be lined up. Fortunately for us, though, this happens in the cerebral cortex. And the reason this is particularly good or exciting in terms of studying brain activity in man is the fact that the cerebral cortex is the part of the brain that has evolved the most through evolution. So it's the parts on the surface, the outside of the head, that's sort of curled and folded, and that's the part to which MEG and EEG are most sensitive. To put it a little bit more physically speaking, if you were to zoom up on one of these sort of, well, this is a sulcus inside the brain. So here you have sort of the outside surface of one of these edges. This is where all the synapses are located. And you have these dendritic trunks that lead to the pyramidal neuron bodies, cell bodies. And when you get semi-synchronous activity, these synapses induce currents in these dendritic trunks that travel along and in parallel and synchronously. And these postsynaptic currents are large enough to produce a measurable magnetic field outside of the head. What's important to point out, though, is that action potentials, for example, the things that most people think of when they think of neurocommunication or signals that are traveling between neurons, the actual action potentials do not produce magnetic fields outside of the brain. But then we come back to, what does this mean when I'm talking about measurable? Well, if you look at a logarithmic scale of magnetic fields, we live somewhere here with Earth's fields. An MR system, for example, produces magnetic fields that would be somewhere up here on the order of 1 Tesla. If you go many orders of magnitude down lower, you start to see that in the pico and sort of femtotesla regime, that's where you start to see magnetic fields that are strong enough to be produced by the brain that can be sensed outside of it. And fortunately for us, there's this great technology called SQUIDs that have sensitivity on the order of some femtotesla, which means a SQUID can be used to measure signals here. So long story short, that means everything in this sort of range is measurable. Yeah? What do MEG systems look like today? Well, as I showed before, it's a helmet with an array of sensors. So this is no helmet, but just the layout of the sensors themselves. This is an Elekta system, or Elekta layout, let's say, with 102 chips like this surrounding the brain. And on each of these chips, you have 3 sensors. You have a magnetometer and 2 gradiometers. They're housed inside this large helium dewar. So the bulk of this hardware, let's say, is really just to house the coolant needed to keep these sensors as cold as they are. And just some quick facts. A full system costs something like 40 million crowns. You could probably get one for maybe 20 if you're good with negotiating with Elekta, or you don't buy all the bells and whistles, but it's on that order of magnitude at least. The liquid helium consumption is something you can't really get away from. So here in Sweden, Daniel has told me it's something like 500,000 crowns per year, but I can guess that price is going to be going up in a year or two. So this is a cost that you essentially always have. There have been developments to try to make recyclers for this helium consumption that would remove or at least significantly reduce this cost, but they're not commercially available yet. One more thing to point out is that the sensor to scalp separation, so the distance between these sensors and the surface of the head, are 18 millimeters at best. I'll talk about that more momentarily, but you can actually kind of see that this is very much a marketing picture. The head in this picture is indeed too big, or the sensor array should be made bigger, because these sensors are each about 20 millimeters, and if you try to scale that to the distance of the head, it looks like it's less than a centimeter or so. So this is a bit of cheating on the Elekta part, but we'll cough that up to the marketing.
Sorry, question.
Yeah, sure.
Compared with the clinical MRI equipment, what do you have there for? I mean,
It's the same.
it's the same basically.
Sorry, clinical MR.
Yeah.
For cost.
For maintenance, yeah, cost.
That's a good question. So it's roughly the same, but a significant part of the cost for MR systems actually isn't the hardware itself. It's the infrastructure you have to build into the ground to support these tons of metal that are used, or the superconducting coils, in fact, that are used in MR systems. But they do consume helium, not quite at the same rate as MEG systems, but they also do, yeah, consume helium as well. That's a good question. So then, why in general would one try to do MEG with high TC squids? Well, the bulk of this talk I'm going to be trying to convince you of the possibility that high TC squid-based MEG can be better than standard MEG, or the commercial MEG systems that are available today. And the reasons I'm going to try to highlight are that high TC squids can demonstrate improved sensitivity to superficial or sources that are close to the outside of the head. Because of the fact that they're closer, you can improve the spatial sampling in the magnetic field around the head and gain in that way, and therefore improve resolution. And also, something that Dag has just pointed out to me that I never thought about before is the fact that because the cooling systems can be made more compact, you can reduce the cable length basically between the amplifiers and the squids themselves, and therefore improve the bandwidth quite a lot. It's not really clear if this has any real clinical and neuroscience applications yet, because nobody's really looked for megahertz frequency type signals inside the brain, but it's not unreasonable to say there might be something there. Finally, though, or some other sort of side benefits, I would say, is the fact that it's going to be a lot cheaper. So we would be using liquid nitrogen cooling, and roughly 80% of the atmosphere is liquid nitrogen, or is not liquid, thank God. It's gaseous nitrogen, but you just need to basically compress it to make it into a liquid. So you have lots of, you have a very abundant source of this coolant. Whereas liquid helium is scarce. It's actually a finite natural resource, and it gets more expensive as time goes on. And finally, just the fact that we're cooling to higher temperatures than the low TC systems provides the capability to have a much simpler cooling system in general. So to start trying to hammer home my message that high TC squid-based MEG can be better than that which is available today, I'll start with a note on insulation. So if you're looking at biomagnetism, so this is basically any magnetic field that's produced by some biological system, for example, the heartbeat, magnetic nanoparticles, or brain activity, life is happening at 300 Kelvin. Yeah? So if you look at a distance scale, so here you have 300 Kelvin where biology likes to live, and here you have a distance from that sort of biological system. And if we consider these to be the sources of the biomagnetic fields, then the standoff distance gives you an idea of how the magnetic field strength will be for different squid technologies. So if you look, for example, at a high TC squid, because of the fact that we're only cooling to 77 Kelvin, means that all you really need to separate the squid itself from this room temperature environment is a vacuum and a fairly strong window. And that means the distance between these warm sources and the cold sensor, when it's operating, can be reduced to less than a millimeter, in fact. In our lab, I think our record is somewhere around 300 or 400 microns. So you can bring the squid, while it's still functioning and superconducting, very close to the room temperature environment. If you compare that to low TC squids, because of the fact that they're operating at 4.2 Kelvin, so much closer to absolute zero, you need a lot more insulation, or you basically need to be boiling off a lot more helium. So the systems, the MEG systems today, have been engineered with a trade-off between how much helium they're going to burn off and how close the sensors can be to room temperature. And typically what you see is that the sensors inside a full head MEG system, for example, require something like a few centimeters of standoff. So you need some few centimeters of insulation, including a dewar, vacuum space, and some super insulation stuff in between your room temperature environment and the sensor itself. To put a very hand-wavy graph on here, magnetic fields, so if this is where your source is, magnetic fields decay as something as 1 over r to the n, where r is a distance from the 300 Kelvin source. Yeah? So this n can be squared or cubed or higher order, but it basically means it's decaying. And if you compare then the magnetic field strength as recorded by a high TC squid as opposed to a low TC squid, if they're looking at the same source, you can see a gain in the signal strength on the order of 10 to perhaps even 100. And that means the signal levels basically you're receiving from your sources are going to be much higher for high TC MEG. However, to understand that better, I think it's important to try to take a look at what is it that we're actually looking at when we're looking at magnetic fields produced by the brain. So to do that, what people often do is they start with this sort of spherical head brain model. So here we have the brain, inside which you have a set of electrical currents that are sort of simulating brain activity. And then you have the skull, scalp, and cerebral spinal fluid and everything else that adds roughly another centimeter or so before you get to the outside, the room temperature, yeah, well, basically outside of the head, right? So we're non-invasive. Our sensors are going to be somewhere out here. And with a high TC squid, as I mentioned before, because they're warmer, they can be cooled with a simpler technology. They can be brought very close to this room temperature environment, that is the surface of the head. You can get a spacing something on the order of 1 millimeter, whereas the low TC squids are sitting some few centimeters away. In this particular simulation, I'm also using different sizes of squids, but that's sort of a practical issue that I won't go into too much detail about. If you then sort of tilt this brain by 90 degrees, so it's tilted down in front of you, and you look at the lead field patterns for the two squids. So here we have our high TC squid. As I said, it's smaller and closer to the head. And the low TC squid, it's larger and a bit further away. The lead field pattern is the sensitivity pattern of the currents that would be coupling to this particular squid. So when you look at one squid, you would see, for example, that the best coupling you have to this squid would be dipoles that are pointed upward here, to the left here, downward there, etc. Basically, dipoles that are circulating the squid in a radius somewhere, something like this, yeah? So this sensitivity field pattern, you can see, is much tighter for the high TC squid than it is for the low TC squid. And this tightness, first of all, brings the fact that the magnetic fields are more, or the spatial resolution can be improved, but it also leads to larger signals. Oops, sorry. So to make things even more complicated, if you take a slice through this brain, each of these brains, and you look at it again from the side, you can look at what are called the isocontour fields. So here I have the two squids, low and high TC squid. This is the brain, and this is the head around it. You take a slice through it, and you get a lead field pattern that looks something like this. So each of these lines is an isocontour representing how strongly a given dipole would couple to each of these squids. And basically what you can see, for example, is if you have a dipole here on this blue line on the surface of the scalp, it would couple roughly 20 femtotesla to the squid if its strength is 1 nanoamp meter. Similarly, for the low TC squid, that position isn't quite the same, but yeah, that's the isocontour for it. If you then follow the surface of the brain and use this parameter phi for the angle between the axis of the squid and the location of the dipole on the surface of the brain, starting at 0 and going through pi, you get curves that look something like this. So again, here we have phi going from 0 down to pi, so starting at 0 to pi. And you have the strength of the coupling between a given current source, so let's say 1 nanoamp meter, and you get the strength of the field that the sensor will see for that given source. So these are the same sources but different squids. Because the high TC squids are so much closer, you see that you get a gain of a factor of roughly 8 for very shallow sources in the signal strength when you compare low and high TC squids. But just one sensor doesn't give you MEG. To do MEG, you need an array. And what you can do there is formulate a theoretical array. So this is, again, something of an abstraction. So I placed sensors in sort of a spiral array around the head for both low and high TC squids. You can see that the packing of the high TC squids looks a little bit more dense because of the fact that they're smaller. You also, for this particular simulation, you will include the noise factors for the sensors. So for standard high TC squids that we produce in our lab, we typically reach about 50 femtotesla. We know that this number can be pushed down quite a bit further, but just to keep things sort of fair, I'm using a fairly high noise level, let's say for the high TC squids. The low TC squids, I use a sort of standard 3 femtotesla noise level. I also use the same edge-to-edge spacing for the sensors, but because the sensors are different sizes, in fact, I can pack 286 high TC sensors onto this scalp surface, whereas I pack only about 109. So this is quite similar to the layout or equivalent, let's say, to that which is used in standard MEG systems. But as you can see, because these are larger sensors, they cover about 35% of the solid angle of the whole sphere. So in other words, if you're just looking at the top half, you get roughly 70% of the sort of head coverage for the low TC SQUID array, whereas the high TC SQUID array only covers about 40%. So another way of putting that is that the high TC SQUIDs in this array are not optimally placed. They're too far apart from one another. They can be packed more densely and therefore record more of the activity coming from the brain. But anyway, in the confines of this simulation, what you can do is look at the power signal to noise ratio per channel in these two arrays. So if you start by looking at the low TC SQUIDs, you see that the first few channels in this is orthogonalized, don't worry too much about that, but the first few channels in this sort of theoretical array receive a signal to noise ratio on the order of 10 dB, or that's roughly a factor of 10 in power. Yeah? The high TC SQUIDs are not as good, right? And that is actually coming from the fact that high TC SQUIDs have higher noise levels. So I'm using 50 femtotesla per root hertz for the high TC SQUIDs, and more than a factor of 10 better noise levels for the low TC SQUIDs, right? So that means the first few sensors of the low TC system are far better than or the first few channels, I should say, are far better than those of the high TC array. However, the signal to noise ratio drops rather precipitously down to the point where the power signal to noise ratio reaches 0 dB or 1. So this is the area where the signal is roughly equal to that of the noise. The high TC array, however, has a longer tail. And another way of formulating that, and perhaps a more illustrative way, is to look at the actual total information capacity in bits. So this is how much information you're extracting from the brain for one sample of the brain activity with each of these sensor arrays. When you do that comparison, you find that the low TC SQUIDs start out good, so each channel you add adds a lot more information, but they sort of flatten out somewhere around 300 bits per sample. Yeah? The high TC SQUID array, however, continues to increase. And again, this is because of the longer tail in the signal to noise ratio. Yeah?
Can you explain what you mean by number of orthogonalized channels?
Sure. So one of the, this is a diversion, and I apologize for those of you that don't know MEG terminology so well, but I'll try to explain it fairly quickly. Basically, the problem with this type of array when you're trying to look at information is that if you're sampling a system redundantly, then you get two channels that receive, let's say, the same information. And that doesn't necessarily improve the amount of information you're getting, right? You're just getting redundant information. That's the case for all MEG systems. So because of the lead field patterns overlapping between adjacent sensors and basically in clusters of sensors around each other, each sensor is sensitive to currents to which many other sensors are also sensitive, right? And you therefore have to orthogonalize the lead field patterns. So you do a transformation of the lead fields such that each channel is then independent. And that basically means you take a linear sum of several of these channels, and you know that the linear sum of this first orthogonalized channel is independent of the lead field pattern of another channel that you're adding to the system. Did that answer your question?
More or less.
Okay. Yeah, I mean, basically what you need to do is separate the channels from one another. So to do that, you take arbitrary linear sums, let's say, of each of the channels, and you add and subtract them to make them independent of one another.
Is that a transform, or is it?
Yes, it's just a transformation.
Okay.
Yeah. Okay, so that was MEG with high TC SQUIDs. Hopefully I've given you an idea of why we might want to pursue that. The next thing in my talk is to explain a bit about what we've already done with our high TC SQUIDs in terms of MEG recordings. So Fredrik Oijland, who is here in the audience, you can wave for everybody. He was one of the pioneers, let's say, of high TC SQUID-based MEG recordings here in Gothenburg. Again, thanks to the MEG MRI project. What we had was a cryostat with a SQUID on top of it, and as I mentioned, the SQUID can be very close to the room temperature environment. We put it up to the back of Fredrik's head and we tried recording. But thanks to the collaboration with MedTech West and people from the Gothenburg University and the Karolinska University Hospital, they gave us very good ideas for what types of signals we should be looking for inside Fredrik's brain. So the first thing we did was to look for what's called alpha. And alpha is a kind of special signal for neurophysiologists because it was the first signal that was recorded from, the first electrical signal, I should say, recorded from live brains. It's a so-called resting state rhythm. And it's strongest from the visual cortex, which is why we place the cryostat at the back of Frederick's head. And you can imagine there's sort of a set of independent sources that oscillate at frequencies around 10 hertz. And this set of sources, when you're receiving visual inputs, they're sort of ignoring each other. So they're oscillating, but they have no phase or frequency locking, which means you don't get much of a signal at this frequency. However, when you close your eyes, these sources sort of couple to one another, and they start locking phase and frequency, and you therefore get a strong signal at roughly 10 hertz. So we put the cryostat, as I said, up to Frederick's head. He started out with his eyes shut, and then opened them at 32 seconds, and this is the trace we got, which more or less looks like a lot of noise, but you can already get a sort of hint of what's going on. There's a signal here that's showing up, and then when he opens his eyes, it's attenuated, yeah? Looking at a spectrogram, I think, is a nice way of making this a more visually appealing presentation of this data. So between 10 and 12 hertz, Frederick has a nice alpha oscillation. So these bright spots represent this strong signal in the spectrogram of, something around 10.5 hertz, for example, here. And you see that the signal sort of comes and goes, but it's fairly strong, so on the order of some fraction of a picotesla. And when he closes his eyes, that signal is significantly attenuated, yeah? Well, we didn't stop there. So the next signal we thought we'd try looking for is what's called the somatosensory alpha, or the mu rhythm. This is a signal similar to the alpha rhythm that's attenuated when you're receiving somatosensory inputs. So if you're feeling some stimulus on your hands or other parts of your body, or if you're controlling them in some way, these signals are attenuated. But when you're not receiving somatosensory input and you're not providing motor control for your hands, for example, then the signal is stronger. We then placed another squid. So we had one at the back of Frederick's head. We had another one placed here over the hand area of the somatosensory cortex for Frederick. We did the same type of experiment, but this time we had two channels. So Lars Jonsson here at Chalmers is very skilled with his woodworking, in fact, and built a nice non-magnetic stand for these two cryostats that you see pictured here. And Frederick sat down and we placed the cryostats not like this, but something like this, yeah? So I have a little quiz for everybody to try to wake everyone up. I can imagine it's after lunchtime. This is already the second talk, and I'm not a very exciting speaker, so let's see if I can wake you up a bit. So just to remind everybody, when Frederick has his eyes closed or he's not receiving any kind of somatosensory input, he has a strong alpha or mu rhythm that's attenuated when he opens his eyes. This can be pictured in this spectrogram. So what I'm going to call this first area is a strong signal. And when, his eyes are open, it becomes weak, right? So keep that in mind. Strong means you have lots of bright spots, lots of activity. Weak, no activity, yeah? What results do we expect? So if Frederick starts with his eyes open and his hands relaxed, after 32 seconds, he then shuts his eyes and flexes his hands. What do you expect to see in A and B? Do you expect to see strong and then weak? Or weak and then strong, yeah? So strong and weak are 1 and 2, weak and strong are 3 and 4.
The visual area.
This is for the visual area for O2. Simultaneously record the hand area for C4. So similarly, do you expect to see strong here and weak here, or weak here and strong here? So I'll give everybody a minute or so to think about their answer. And once you have your answer, I'd like you to close your eyes so that we can vote, and there will be no cheating. So when you have your answer, please just close your eyes. Okay, so not everybody's closing their eyes, but I'll ask you all to close your eyes now. If you don't feel like voting, that's okay. But close your eyes and raise your hand with either 1, 2, 3, or 4 fingers up for which of the 4 answers you think is correct. Okay, so lots of you are answering. That's good. You can put your hands down and open your eyes again. There's a bit of a mix. So just to keep things going quickly, I'll give the answer. So it's 4, yeah? Many people said that, but just to specify a little bit more clearly, Frederick started with his eyes open. That means he's receiving visual input. The alpha sources are not locking frequency and phase, so you have a weak signal. When he closes his eyes, they lock frequency and phase, and you get a strong signal. Similarly, when he has his hands relaxed, when he has his hands relaxed, he's not receiving somatosensory inputs, nor is he sending motor control signals to his hands. So you get nice, strong mu rhythm signals there. And when he flexes his hands, he's getting both somatosensory inputs as well as providing motor control. So that signal is attenuated. Okay, that didn't really wake everybody up, but let's keep moving anyway. So again, to come back to this, why bother with high TC squids? Talking about this gain term in terms of coming closer to the head, what we wanted to do is try to investigate that with some real measurements with MEG. So what we did was, as Daniel mentioned, Minshu, Alex, me, and Matti, and Robert, and others got together at Karolinska at the NatMEG lab, and we set up our wooden structure for holding the cryostat. And what we aimed to do was compare a sensor inside this system with one inside here, placed at sort of the optimal positions for different types of stimuli. So obviously this system is capable of full head mapping, but we're picking one sensor, the sort of best sensor inside this MEG helmet, and comparing the signals that are recorded by the high TC squid at the best position for that particular source. So when you do a full head map, starting with auditory evoked fields, so this is just a simple tone. Thanks to Daniel, we had good guidance on how to generate this particular stimulus. You just apply a tone at 0 seconds, and some 100 milliseconds later, you get a nice response from the auditory cortex of this subject. We then look carefully at this response at about 100 milliseconds, and we want to try to localize that source. And we can do that by looking at the topography. So this is now just one slice of this data, looking at all of the low-T MEG channels in the NatMEG system. And you get a field map that looks something like this. We were focusing on the right-hand side of the brain. So here you have a source that's somewhere here, pointing down towards the right ear. That's producing, let's see if I'm doing, yes, producing an inward-pointing magnetic field here and an outward-pointing magnetic field here. We then source localized this particular source to know where in the brain it's coming from with the M&E and field trip softwares. And we predicted where on the head the field would be maximal for which we should record with the high TC squids. And when we do that, we get traces that look something like this. So if you compare the low TC channel, which is this blue trace, for the positive peak and the negative peak, you see that you get a signal something on the order of 200 femtotesla. If you then use a model to figure out what field magnitude you would expect by coming closer to the head, as I keep trying to say, when you get closer, the magnetic fields get stronger. So you can use a model to predict how much larger that signal should be by coming closer, and you get a factor of between 2.5 and 2.8 gain in the signal strength. So that's what these blue and, or sorry, this pinkish and red curves are. These are just scaled up versions of this low TC trace, both for the positive and negative. And you see that the high TC squid magnitude roughly matches what you would expect to see for the dipole field. So we can show with these traces more or less that things are behaving as we expect them to. By coming closer to the brain, the signals get stronger, in this case by a factor of almost 3. We also looked at another set of sources, and these are the so-called somatosensory evoked fields. So instead of providing an auditory tone, we actually applied a strong electrical stimulus to Robert's median nerve. And we did that many, many times. And this is the trace that you get for the low TC squids. And we then tried to look at this 20 millisecond delayed first response, you can sort of call it, to this stimulus. So this is delayed by roughly 20 milliseconds because of the fact that there's a finite length between the stimulus position and the brain. So the signals take roughly 20 milliseconds because they're traveling roughly 50 meters per second in the body. Again, we look at the field map over the head. We localize the sources and predict what the high TC squid signal should be. And there you get these pink and red curves for the negative peak and the pink and red curves for the positive peak. However, as you can see, the high TC trace does not match that at all. It's not really clear to us yet whether this is an artifact or not. It certainly could be, so we're not ready to claim anything groundbreaking about this yet. We basically are going to try mapping this field pattern better with the high TC squid to try to understand better what this type of source could be. But yeah, essentially speaking, the signals that we expect to see from this kind of source are not those that we record with the high TC squids. So that's a fairly rough overview of our past achievements with high TC squid-based MEG. And now I'd like to talk a little bit about our future hopes and what we plan on doing with this big NeuroSQUID project. Any questions so far? No? Good. Nobody's awake. So we've developed a consortium. We're calling the NeuroSQUID Consortium. It's headed by Chalmers. And the Chalmers development areas are going to be in nanoscience and high TC superconductivity, so investigating materials and sensors with the advanced high TC superconducting investigations. We also are going to be working on validating our high TC squid-based MEG systems with colleagues at GU and Karolinska, Martin Ingvar and others at the NatMEG lab. The general idea is to try to see if we can use these sort of high TC nanosquid structures to image brain activity in the ways I've shown already and study interesting neurophysiological signals or psychiatric disorders, etc., with such a full head imaging system. So the first part, exploring high TC materials at the nanoscale. This is work very much coming from Thilo Bauch and others here at Chalmers. They've been working for years on investigating superconductivity in high TC materials, basically trying to understand the physics, the fundamental reasons for high TC superconductivity. That's still one of the very big mysteries in physics. They've developed this very nice technology for producing very robust and reproducible nanosquid or nanojunctions in these high TC materials. So this is a very big challenge in this particular technology because of the fact that high TC materials are complicated materials. They're in fact perovskite oxides, so they don't grow like metals. They have to be grown in very special conditions, and fabricating them at this sort of scale is an extremely large technical challenge. But what they've been doing previously is exploring these materials at the nanoscale, and they aim to test theories that predict, for example, why these materials go superconducting and how. And they'd also like to investigate the Josephson-like behavior of these nanowire links. So what's interesting for us in this context is the fact that these bridges behave as Josephson-like sort of junctions. And those of you that know about squids know that the Josephson junction is really the heart of a squid. So the entire phenomenology with which squids operate is based on the fact that you have Josephson junctions in these superconducting materials. But what's particularly exciting about these structures is the fact that if you make such a nanosquid and you look at the noise characteristics here of such a system, you see that, for example, if we do that for our standard high TC squids, you get a noise level that's about here. So it's between a microphi-naught and a phi-naught. Don't worry too much about these numbers, but it's roughly here. And with the nanosquids, while they have fairly high 1/f noise that will be investigated, of course, but you can see that at higher frequencies, their noise levels actually approach pretty much the electronics background. So possibly this noise level could go well below the microphi-naught range, which would mean if you were to be able to fabricate such a squid or a larger squid based on this technology that's similar to our high TC squids, you would be able to reach sub-femtotesla sensitivity with these high TC nanosquids. So this is a particularly exciting approach for us to be able to improve the sensitivity of our squids and therefore really beat low TC squids when it comes to MEG recordings. But nowadays we're sticking to our typical workhorse. This is a high TC squid fabricated by Fredrik Oyhon many years ago. It's a 10 by 10 millimeter substrate with a roughly 9 millimeter pickup loop. You have a magnetic field that penetrates this loop and causes currents to travel here, and the squid component that's active, let's say, is just on the micro scale down here. And what we've been doing for our MEG recordings is putting this into one of these cryostats and trying to bring it very close to the room temperature environment. The next step, though, is to basically build new versions of these cryostats that can house more channels. So in this case, we've started with designing a 7-channel system in which the squids themselves are tilted with respect to one another, aiming to match more or less the average curvature of the head. And that way we can start doing multi-channel and MEG recordings of brain activity. This is what the cryostat looks like today. Unfortunately, it's in pieces. I was hoping we'd be able to show it to you guys at this conference, but it is in pieces up in our workshop. Lars Jansson and Staffan have been really critical to putting this thing together, but we're hoping to test it in the next couple of months. So this is a 7-channel cryostat. We also aim to work on validating the technology with neuroimaging studies of various kinds. So I'll highlight just a couple of investigations that some of our colleagues at Gothenburg University are working on developing. So there's been a lot of activity at GU studying what's called, or using a technique called microneurography. And this is a particularly exciting technique because of the fact that you can use electrodes placed inside the nerve bundle inside the arm to address single fiber tracks. So you can actually record activity from a single efferent nerve fiber. So for example, if you touch here on this one little spot on this person's arm, you would get a burst of activity from this receptor that looks something like this. And this is the single action potential, which is just one of these little spikes. What I think is even more cool about this technology is the fact that Johan and others have been able to develop the hardware for this, so the amplifiers and such, that they can actually stimulate with these electrodes as well. So instead of pressing on this receptive field, they can send a signal that sort of emulates being touched here or somewhere on the hand. The brain receives the signal in the same way as it would have if it were from the receptive field and interprets it in the same way. But it's truly an artificial signal that you're sending in. With this technology, combining it with MEG, as Johan has already made a lot of progress so far, using the brush robot to study different types of touch paradigms, they aim to do both recordings with a microneurography as well as stimulation to try to see if they can see the brain response to a single efferent input. And finally, I won't get into too much detail about this, but we've also initiated a potential EU collaboration. We submitted the proposal a few months ago. It's involving a very large network of different groups. Mikkel is here somewhere. Yes, there. From ULISH, Mikkel and others, I should say, at ULISH have nice SQUIDs that they've already been doing similar experiments as we have to compare their high-TC SQUIDs with low-TC systems. And Elekta, of course, is the company that's making the commercial MEG systems today. They're on board, as well as other partners, including a group from Twente that have been working on microcryocooling technology. I won't get into too much detail about that. But what's particularly exciting about their technology is the fact that we can develop an even better MEG system than that which we can do with the standard doers that we're working on now. So just to compare a standard MEG system today, you have a spacing, as I mentioned before, on the order of 20 to 30 millimeters. So the distance between a sensor and the head surface is 20 to 30 millimeters. This is sort of our dream or our hopes for high-TC SQUID-based MEG. You could have an array of these sort of micro-cryocooling systems that would house a set of sensors that would fit snugly and close to an arbitrary head size or shape. So such a system could be used, obviously, for improved studies on adults because of the fact that the sensors are already closer to the head. But instead of having to build an entirely new MEG system for recording on children, this would be flexible enough that it could be used for children's studies as well. And the real goal, yeah, sure.
To achieve in this setup then?
Sorry?
You talk about the improved on sensitivity.
Yeah.
What do you mean, like temporal resolution or?
No, not so temporal resolution doesn't change. Yeah, I won't say anything about that, let's say. Okay. It's really the spatial resolution that we're aiming to improve. And perhaps to put it another way, the neuromagnetic field, as it's radiating from the head, as I've said, it's decaying, but also it's the spatial frequencies are the spatial frequencies to which you're sensitive at some distance from the head are limited from the fact that you're far away, and by sampling more densely around the head, you don't gain more information about the brain. But by coming closer, we can pack the sensors more densely, and we know that we can gain more information with each channel, let's say, that's added, and therefore improve the spatial resolution as well. Yeah? So, yeah, that's part of the motivation. Yeah, definitely. Yeah. So yeah, to restate what I just mentioned, our real aim or the long-term aim of this project would be really to push the spatial resolution down to something closer to what you get with fMRI. yeah, this is just a sort of schematic. So we have the sensors from Chalmers, ULISH, Avon, and VTT. Juho is here. He just received his PhD developing a KIM technology, which is another potentially very exciting could be high TC SQUID-based magnetometer technology. We'll have microcryocoolers to develop an array like this that would be optimized by theory people working from Aalto, PTB, and ULISH. That would be integrated into a full head system with which you would need imaging software. Again, you'd have to you'd have to rework a lot of the ways in which source localization is performed because of the fact that this is a moving sensor array. And we aim to validate it with people from Italy, Karolinska, obviously NatMEG, and other groups. So that's our future hopes for NeuroSQUID. I've given you a very rough overview of why you might want to pursue MEG with high TC SQUIDs. I've talked about what we've done before, as well as explaining what we hope to do in the next 5 years or so. But of course, this work isn't just mine. So this is a nice picture taken from our trip, one of my first trips, I believe, to Stockholm, in fact, where we did recordings with the benchmarking, comparing different technologies, our high TC SQUIDs versus the low TC SQUIDs inside the NatMEG system. So Min Shu is a PhD student here at Chalmers. Daniel, you've already met. Martin is the one, as he said, responsible for this whole system. Stephen was a big help on these recordings, as well as with a lot of the analysis. And then, of course, we have some of the fathers of MEG, which spoil us with their knowledge. So it's been a great collaboration so far, and we're looking forward to more. And then more locally, there's been a lot of different people active on these high TC SQUID-based MEG recordings. As I mentioned before, we had smart people at the Karolinska University Hospital telling us what kind of signals we want to look for. I'm sorry, I should have included a picture of Elena and Bushra's there, but I don't have a picture of Elena. But Elena, maybe you can wave. She's been also pivotal in the developments that we're doing more recently. And we have Min Shu, Bushra, Frederick, as I mentioned before, and Joran, who helped a lot with the measurements. So this is Frederick getting wired up with the EEG electrodes for some of the recordings we did with our high TC SQUIDs. Dag has been the PI here at Chalmers, and Mikkel the one at GU. And then Maxime and Alexei are some of the guys here locally that really make everything happen. So thanks to them, and thank you all for your patience and listening.